cell division. For the first subtopic 3.1, the cell cycle, at the end of the lesson, students should be able to show the stages of the cell cycle. Modern cell theory states that all new cells are derived from other cells. It enables all organisms to grow and reproduce again. Based on Campbell 9 edition, cell division means the reproduction of cells. It requires the distribution of identical genetic material, which is the DNA, from the parent cells to daughter cells, except for meiosis. The cell division process is an integral part of the cell cycle, the life of a cell from the time it is first formed from a dividing parent cell until its own division into two cells. There is two basic types of cell division, which is mitosis and meiosis. For mitosis, it is a process of nuclear division in eukaryotic cell and consists of four phases. The first phase is prophase, followed by metaphase, anaphase, and the last one is telophase. The result of mitosis is all daughter cells having the same number of chromosomes as the parent cell. For meiosis, it is a type of cell divisions that occur in germ cells of reproductive organ. Reproductive organ in animals refer to testes for male and ovary for female, while the reproductive organ in plant is stamen for male and carpel for the female. Meiosis consists of meiosis I and meiosis II, which result in all daughter cells having only half the number of chromosomes found in the parent cell. Before the cell can divide, the DNA, which is the genetic material, must be replicated. Then, the two copies of the DNA will separate, so that each daughter cell ends up with a complete set of genome. Now we continue with 3.1, the cell cycle. Cell cycle is the complete sequence of events in the life of an individual diploid cell. The cell cycle consists of two main phases. The first phase is interphase, which refers to the non-dividing phase or the resting phase, while the mitotic phase refers to the dividing phase. The M phase includes mitosis and cytokinesis. Mitosis is the division of the nucleus, while the cytokinesis is the division of the cytoplasms. Cell cycle can be divided into two phases, which is interphase and mitotic phase. In interphase, there is three subphases, which is G1 phase, S phase, and G2 phase, while in mitotic phase, there is two subphases, which is mitosis followed by cytokinesis. Interphase. Interphase is the longest phase in the cell cycle. It takes about 90% of the cell cycle, and it is the phases or the stage before the cell can enter the mitotic phase. During interphase, chromosomes are in the form of chromatin and are invisible under light microscope. During interphase, the cell grows by synthesis protein and cytoplasmic organelles such as mitochondria and endoplasmic reticulum, and duplication of chromosomes occur for the preparation of the cell division. Interphase can be divided into three subphases, which is G1 phase, S phase, and G2 phase. For G1, the cell grows before the DNA replication occurs during the S phase, while for G2 phase, the preparation for cell division occurs. During mitotic phase, the cell growth will stop and the cell is ready for the cell divisions. Cellular energy is focused on orderly divisions into two daughter cells. Mitotic phase can be divided into mitosis and cytokinesis. Mitosis, which is the nuclear division, consists of four phases, which is prophase, metaphase, anaphase, and telophase. At the end of telophase, the division will be followed by cytoplasmic division called cytokinesis. Mitosis is the division of genetic materials in the nucleus followed by cytokinesis. As the result, two daughter cells having the same number of chromosomes as a parent cell will be formed. After nuclear division, the cell division will continue with cytokinesis, which is the cytoplasmic division. There is two types of cytoplasmic divisions based on the type of cell. In animal cell, the cytokinesis is known as the cleavage furrow, while in plant cell, there is a formation of cell plate. The first phase in mitosis is prophase. During prophase, chromatin will be shortened and thickened, and each duplicated chromosome appear as two identical sister chromatids joined at the centromere. The second phase in mitosis is metaphase. During metaphase, chromosomes as a sister chromatid align at the metaphase plate. The third phase in mitosis is anaphase. During anaphase, the sister chromatid will separate and each daughter chromosome will move to the opposite pole. The last phase in mitosis is telophase. During telophase, the chromosome reach the opposite poles and the chromosome become less condensed, which are uncoiled and lengthened and thus become invisible again.